The second part of Lab 38 covers the phylum Annelida, which is one of the taxa you need to know. Be sure and review the list of taxa you need to know from this lab exercise 38 for the mollusks and now for the annelids. These annelids are commonly called segmented worms, and you can see why in the picture on the right. This is an earthworm, and you can see that the body looks like it's in these repeating segment type there. And so the earthworm is an example of an annelid. Leeches are examples of annelids, and then there's many marine type species like a clamworm, which we'll see in a little bit. So what is a feature characteristic used to distinguish three classes? And so we're going to be dealing with three classes here of the phylum Annelida. And this is one of the distinguishing features is these bristle hair-like structures that they have along their body. And earthworms are going to belong to a group that's called Oligochaeta. And oligos means few. So they do have a few of these bristle-like structures, and they do help the earthworm get traction as they move through soil. And then we have the class Polychaeta. Poly means many, and these are very pronounced in this one. You'll see what I mean in a little bit. The polychaetes, the bristles are much larger and may have even more function than just providing traction. And then the last class has very little bristle-like structures, and the internal structures inside it's not as segmented. These segments do have repeating patterns, but it's called Hirudinea, and these are the leeches. And the leeches, the Hirudinea, they get their name based on a protein they produce in their saliva. These guys are ectoparasites, and looks like this is an eye. They get their name from there's a protein in there that prevents blood from clotting. So when they latch onto an animal and are sucking the blood off of the animal, this protein helps prevent the blood from clotting, so it flows more easily. So looking at the first group is the Polychaeta, and the first thing you notice is the very pronounced structures along the lateral or the sides of this animal. You can see the segments on there, but they have very pronounced and very many bristles, so that's where Polychaeta comes from. Polychaetes are marine, and I already mentioned what polychaete means, and that means they live in oceans and bays and salt water. They have these lobe-like structures called parapodia, and you can sort of see if you go near the base here, there's this little lobe-like feature. It's more pronounced here in this diagram, gives us an ideal picture of it. So this is a diagram of the anterior head end of this polychaete. And those parapodia are these little fleshy-like extensions, and they do have some functions on there. The bristles on there may have sensory receptors on there, and the lobes themselves provide more surface area that allows the animal to exchange gases with the water, so they take in oxygen and so basically through the epidermis they can do this exchange. Now I mentioned the next question, how are the setae used? The setae are the hair-like structures, bristle-like structures, and so they can have chemoreception properties on there. So the clamworm belongs to the genus Nereis, which you can see there, and I do have a link in that document I prepared that accompanies the lab, and that's what this PowerPoint is based on. So if you click on that, it'll take you to its images similar to this, so you can look at them. We do have specimens preserved in the lab, don't have live ones now. When you look at the head region, the anterior region, you notice that they'll have these tentacles. They'll have a couple of pairs of eyes there, and then they'll have this jaw-like structure here. And this jaw-like structure is actually protruding right now. It's tucked inside, so that's why it says everted pharynx. The pharynx is the back of the throat, so these jaws are actually tucked in the head. In fact, you can see them tucked in this actual image there. They're not protruding as much as you see in the diagram on the bottom picture. in the procedure, if we had specimens for you to mess around with, they ask you to take forceps and pull those jaws out, try to extend them outward. And then this is a cross section, so of the Polychaeta Nereis, and they wanted you to actually look at a parapodium, which is singular for all the parapodia. But I provided a cross section through the entire body to get a sense of what it was we were looking at. So if you can imagine coming back here to this picture, the diagram below, and if we took a section through the body, a cross section right through the parapodia, just like that, that's what the image on the top is. So there's a cross section there. You can see these muscles associated with movement of the animal. You're going to have a dorsal and ventral blood vessel, and that reminds me of the characteristic of these earthworms. We mentioned them earlier in the prior one, and since this is a separate video on itself, let's review that. They have a true body cavity, so they have a coelom, and they have a closed circulatory system, which means their blood is kept within blood vessels. It doesn't get into the cavity or the body cavity. So closed circulatory system. And this was mentioned earlier that mollusks have an open circulatory system except for the cephalopods. They have a closed circulatory system, and the cephalopods, like octopus, are much more more advanced in terms of their complexity and their behaviors and so on. You can see the intestine here in the middle. The space number 3 right here, there's your coelom or your true body cavity. This is the intestine inside number 4. That's the lumen, or the space inside the intestine where digestion occurs. If we move out to the outside, you can see the section right there through a parapodium. So that's it's labeled parapodia plural because they have number 7 on both sides. So you have more than one parapodium is parapodia. So now if we look at the picture down here, this is a parapodium, one parapodium more magnified, and there you can see the setae protruding out the side of the parapodium. Remember, the parapodium is, the epidermis is thin enough where we can exchange carbon dioxide and oxygen with the water. Of course, the animal needs oxygen and produces waste product, carbon dioxide, got to get rid of it. All of the structures that are labeled here are not really necessary to know, just to get a sense of what this kind of structure looks like if you section through one. You can see the bristle-like, hair-like structures, the setae there. This is the other end of the setae, and they have a fancy name for it, the acicula. And those are not names that you need to know. So you should be aware that this is what the parapodium looks like on a prepared slide. There are some other polychaetes, and there are some interesting ones. There's many types. These are just a small little sample here. The first one, you can see an individual holding one of these called a sea mouse. You can get an idea of why it's called a sea mouse, because the setae makes it look like it's a furry or hairy animal. That's what it looks like out of the water, but in the water, it looks kind of beautiful there. You can see the parapodia sticking out to the side. It has many bristles. That's why it's polychaete. This is what one looks like on its ventral side, and this is the dorsal or the back side of it. And then, the genus for the sea mouse is Aphrodita, which is, there's a link where you can access these images, and then there's the genus there italicized. And then Chaetopterus is Chaetopterus. This is called a parchment worm because they produce this enveloped-like material that they secrete, and the worm is inside of there. So it's kind of like a parchment or an enveloped type of covering there. And I was doing a web search to learn more about these myself, and I clicked on, I included a link to a story. It's an interesting a story about this polychaete, many of them washing up onto a beach, a shoreline. Somebody's describing them in that little article. It's a small article. It's worth taking a look at. So these are 2 other representatives besides Nereis, the clam worms. There's the clam worm, the sea mouse, and the parchment worm, which belongs to the genus Chaetopterus. So here's some questions to answer about the observations so far that we've done together. Why name it common name sea mouse? What are the features? Well, all this, the hair-like bristles make it look like a furry little mouse. Be sure to consider what the functions are of the parapodia and the setae, which I briefly mentioned. So make sure you consult your lab manual to answer those types of questions. for yourself. What is the probable function of the tentacles? We saw those little tentacle structures on the anterior end of the head end of Nereis. So what do you think those might do? I'm thinking maybe something sensory, so think along those lines. And then objective one in these labs we do on animals are always going about what are the significance of the fundamental processes for these animals. And those fundamental processes, again, are for feeding, to feed, to reproduce, and to respond to the environment. And so responding to the environment is a fundamental process. So those parapodia, they have those bristles, the setae, that actually have sensory receptors on there, so they can sense chemoreception and sense chemicals in the water. And what features of Nereis indicate that it is an annelid? Remember, annelids are segmented worms, so that obvious you can see the segments on there. And so now we move on to the class Oligochaeta, which means few bristle-like structures. And then there's a video of an earthworm moving along. So I'm going to try to get that video started so we can see it moving, because one of the things you're supposed to do is observe a living earthworm moving. And there you can see it moving, and this movement is quite different than the roundworms we saw in an earlier lab. The roundworms only have longitudinal muscles, so they can only wiggle side to side because the longitudinal muscles run the length of the body, and all they can do is shorten the muscles, and that causes them to wiggle. But this one, the earthworm, has a fluid-filled body cavity, and that cavity is the fluid in there. That pressure is what these muscles along the body wall, the outer wall of this animal, are pushing on. So they call this a hydrostatic skeleton there. So you saw the movement there, and they have what's called a hydrostatic, and that just means pressure within the walls of a container, right? And this container is the coelom. So it's a hydrostatic skeleton. It's not a bone skeleton, it's a skeleton of fluid. Skeleton. And we have what's circular muscles. The muscle fibers wrap around the body of the animal. And so when those contract, they're going to squeeze down on that fluid and actually push that fluid forward, right? And that causes the fluid to move to the front of the animal. And that causes the front of the animal to move upward or forward through the soil. And then once they move forward, their setae on the outside grips onto the soil. And then what they do is they use their longitudinal muscles to pull their tail end forward. So they're constantly doing these rhythmic contractions, stretching it out forward, and then by pushing with the circular muscles on the fluid, that pushes the animal forward, and then the animal locks into the soil and then uses the longitudinal muscles to bring the tail back. So let's see if we can replay that and notice that kind of movement. So there, where the longitudinal muscles are now pulling the tail end forward, and then again, so you have these repeating cycles. So you push the circular muscles, push the fluid forward, pushing the front end of the animal forward. Then the animal grips onto its where it is, like in this case, the sidewalk here, and then the longitudinal muscles pull the animal, the back end of the animal forward overall. So that's basically going to take care of procedure 38.8, the locomotion of the earthworm. And there is a diagram that shows and explains this, talking about the circular muscles. You can see those strands wrapped around. They're on the outside, and then the longitudinal muscles. So you can see when the circular muscles are pushing on the hydrostatic skeleton, that's going to push the front end or the anterior end where the mouth is of the animal forward. So the animals move forward, and then at this point here, the longitudinal muscles are going to pull on the back end and move that upward in this diagram. And so you have this repeating rhythm of contraction of these muscles back and forth on that hydrostatic skeleton. What does oligochaete mean? We know that means few bristles. The scientific name for a familiar earthworm is the genus Lumbricus. You can get that right out of the lab manual. It's in the background reading there. And the full name of a common type of earthworm is Lumbricus terrestris, like terrestrial, right? And so be sure you understand how the earthworm uses the hydrostatic skeleton and the circular and the longitudinal muscles to produce the movement that you see there in the image. And it is moving forward like bilateral animals tend to do. So that's part of that procedure 38.3, observing that locomotion in this diagram study that does help explain how those muscle action works, the longitudinal and circular muscles. And then there's a question to answer about those observations, does the earthworm look like it's moving randomly? Seems to be a little bit more coordinated of movement. And in this case, when you're watching the earthworm move, what do you think it is looking for as it's moving around, right? It seems to be twisting and going in one direction and moving the other. Maybe able to sense its surrounding is looking for a moist habitat to go into, because the earthworm can dry up if it remains outside too long. What muscles allow the worm to change its length and thickness? So there, the length changes the longitudinal, and the thickness is the circular muscles that push on the fluid. And then you've got to answer a question about how does the movement of earthworm might compare to a snake. Have you ever seen a snake move? But in the prior lab, there is a video that I provided for you that allows you to look at how the nematode wiggles back and forth, right? And then again, it asks what features of Lumbricus, the earthworm, make it an annelid, the obvious segmentation there, right? So another part of this laboratory is supposed to be doing observations of the anatomy of an earthworm and then related to some of the important fundamental processes like reproduction, right, and feeding and responding to the environment. So here, looking at the external anatomy of the earthworm, this would be the anterior end of the animal. And if you had a specimen that we would normally provide in a laboratory situation, if you rub your fingers along the surface, you'll know you're on the belly side when you can feel the little bumps or the setae. They're not very big, but they're big enough for you to feel with the sensitive touch of your fingertips. And then if you put them under a dissectoscope, you can get a better look at those. So here we're actually looking at the ventral side of the animal. And if you move back a certain number of segments, you're going to come to what looks like an area where the segments have sort of fused together. And that area there is called the clitellum. And that's right there. So make sure you look at that, and that's the clitellum. And that structure is an important structure associated with the production of cocoons. These animals, earthworms, are hermaphroditic, so they produce both sperm and egg. And if you look toward the front end here, the very first segment is called the prostomium, and stoma means mouth, right? And so pro means in front of, could mean first, and so first segment, prostomium, and then there's the mouth, a little opening. It's more on the ventral side instead of the dorsal side or in the front. And then the next segment seems to wrap around the mouth, and that's what peristomium means. Peri means around or to surround. And so the prostomium and the peristomium, and then there's the mouth location. So I'm already learning a bit of that external anatomy. And then if you count a certain number of segments, he'll take you to areas where you find certain pores or openings that allow for the transfer of gametes during their reproduction. You can see the segments here. It's pointing to the little setae there. At segment 10 here is where you're going to find these seminal receptacles that receive sperm. Remember, both animals produce sperm, but they don't self-fertilize. They actually cross-fertilize by mating with another hermaphrodite earthworm, another individual. And here, gono is reference to gonad, where the testes or the ovaries are located. And as you're moving further back, this is segment 10, 11, 12, 13. At about segment 14, you're going to have the female gonopore. So this would be where eggs are released through that pore. And then if you go to the next segment, you're going to have the male gonopore where sperm are released, right? So there are some structures that are both external and internal. And internal, the seminal vesicles, but the male gonopores is where the sperm is released. The seminal receptacles is where sperm is received and stored, right? And they'll be stored in the vesicles that are deep in those first pores there in the front. And then you have the female gonopores. And so you can see here, there's a picture of two earthworms in copulation. So they're mating, they're exchanging gametes with each other there. So you can imagine they're going to line up in a way that each one transfers sperm to the other one. And what happens here is the sperm will travel through grooves and get stored, and they'll go into the seminal receptacles and be stored within there so they'll line up relative to these clitella that you see there. and they're able to transfer sperm to the surface of the body of the other, and then the sperm move into the seminal receptacles, seminal vesicles, right? So when the time comes after they've finished mating and they go their separate ways, each earthworm is going to produce this mucousy-like material from the clitellum at the surface, and then with rhythmic contractions causing them to blow up or expand a bit, not explode, but expand a bit, that's going to force that a band of mucousy material forward. And as it's moving forward, first thing it's going to do is going to go over the area where the eggs are released, right here. So the eggs would be released and be picked up by that mucousy membrane, and then move further forward where the sperm was transferred. You don't want your own sperm. You want the sperm that you got from the other animal. And so that band of mucousy material moves forward and then basically comes off the front end of the animal, forming a bubble of gametes in there, eggs and sperms. The sperm fertilizes the egg, and then this cocoon forms, and that cocoon protects those embryos that become eggs. So protects those eggs with the embryos in there as they mature and then eventually hatch to form a larval young earthworms. So that's how they reproduce. So looking further at some of the external anatomy, I already mentioned some of this. You can notice that you have the segments there, and so there are some key segments, like segment number 10 will get you to where the seminal receptacles are. And under the dissecting scope, you can see those pores there. The setae are going to be those bristle-like structures you feel on the ventral side. You notice we already pointed out the mouth there. On the other end, on the back end, they have a complete digestive system. They have a mouth end, they have an anus end where waste comes out the other end. We pointed out the seminal receptacles where sperm from the other individual goes into the female gonopores, male gonopores, and even gave you the purpose of the clitellum. So these structures are described in the lab manual, and they tell you kind of the story of how I told you how they're used to reproduce. And that, by the way, is one of the major functions, or primary functions of animals. You got to leave offspring behind for that next generation, right? Remember, it's feeding, reproduction, and responding to the environment, right? The next section is about the internal anatomy of the earthworms there, and because you're not going to be able to cut one open there at home in this online version of the course, we do have diagrams, one from the lab manual, and then here's a picture of an actual dissected earthworm. And this one is dissected on the bottom along the dorsum. So they kind of opened up the animal from the top and cut into it. And so what you're looking at the internal organs from the inside. And so this is the idea. And we're going to want to make sure that you can at least identify both in the diagram that you see there and in the image of the actual dissection here what you're looking at. If we were in the lab, we also had a large model where you can compare. Models are always nice because they allow you to compare to what you're seeing in a real specimen or in diagrams, which represent two-dimensional models like the one you see here on the top. So some of the structures you want to be able to look at is a muscular pharynx, which is part of the digestive system, the esophagus, the crop, which stores the soil that they take in, and the gizzard is a muscular structure there. And then the rest of the digestive system is the intestine toward the back. The nervous system actually runs along the bottom, so they have a ventral nerve cord, and you can see it labeled in the top diagram. So there's the ventral nerve cord. You won't be able to see it in the dissection because it's underneath those organs, because this is the dorsal side of the organs here. But the nerve cord comes forward and then moves around both sides just in front of the pharynx here. And the brain would be located right up there. So the brain, if we were to look at the dissection here, is about right there, probably outlining part of the brain right there. And then the cords would wrap around it underneath, and then the ventral nerve cord runs along the back there. So the nervous system helps control activities of the body there. So the brain there we identified in the dissection, and you can see it here. This is more of a side view of the organs, so we can see the top and the bottom, whereas this is the top view. So you got to use your imagination to see how would one look and one from one view and then the other. So this is lateral view from the side view, and this is the dorsal view in the dissection there. You should be able to identify the ventral nerve cord, at least in the diagram here. We cannot see it here, it's underneath. And every segment that you're going to see along there, there'll be some specialized regions, for example, of the digestive system and part of the circulatory system. You can see the hearts up here. You should be able to identify the hearts as well, so that's on the list. But for the most part, much of the length of the body is going to have repeating segments of these repeating organs. You're going to see the intestine along most of the segments. You're going to see the nerve cord in all of the segments because it passes through there. And you're going to see excretory organs called nephridia. Nephridium is singular. And every animal, when they're metabolizing, they produce metabolic waste. You got to get rid of it. So the little organ structures that are going to pick up these nitrogenous waste, this metabolic waste from their body fluid, are called nephridia. In a vertebrate kidney, the fundamental units are called nephrons. So there's a similar naming system there to what they do. The other thing you're going to need to know is the dorsal blood vessel. There's a ventral one as well. But since we're looking here at the top, you can see that we have a dorsal blood vessel in the diagram at the top. It's in red. And then a ventral blood vessel. And here towards the front end, this is the anterior, and the rest of the animal is cut off. You don't see the rest of it. It's a longer animal. We don't even get to the clitellum in this picture here. The clitellum is further back here, and then you have the rest of the posterior end of the animal. But you can see that the dorsal, the top blood vessel in this image, and the bottom, the belly side blood vessel, are actually connected by more blood vessels that they collectively call the hearts. Now, they don't really have much ability to contract, but it's more the contraction of the body of the animal that moves and circulates the fluid in there. And that's not unusual because while our heart is pumping blood away from us, the main way blood gets back is through contractions of our muscles. It's not so much to do with pressure, but contraction of the muscles helps push blood back. So changes in pressure on those vessels can get that circulation to move around. So if we look at the specialization in parts of the complete digestive system, you have your mouth up here. The mouth is going to lead to the pharynx, which is the muscular structure there, and then the pharynx in a human would be the back of our oral cavity or mouth cavity. And what follows next is the esophagus. So the green tube that passes through those hearts is the esophagus. Okay, so coming to this picture down here, this first ball-like structure, that's your pharynx. And then here would be where the hearts would be. And underneath there, it's not very visible, but the esophagus would move back through these structures here, including the seminal receptacles that we were mentioning earlier. The pores are underneath there. There were seminal vesicles up here. There were very large structures there. And the digestive system is moving underneath there. The hearts would be located under here. And then eventually, just like we see the hearts here, and then eventually you're going to get to this another pouch-like structure, which is the crop. And like in birds, the crop is going to store some of the freshly eaten soil. And then that soil gets moved back to the gizzard, which is a very muscular organ. If you were dissecting and you were poking around at these organs, the crop feels soft, but the gizzard is really hard. The structure is going to turn that soil and grind it down so it can better digest. And then as you move back further, you get into the intestine. So for the rest of the animals, the intestine, where we're absorbing nutrients from that soil you've eaten. The soil has a lot of rich organic material that can be used as nutritional value for the animal. And so that's essentially taking care of another basic fundamental function, which is feeding. How do you get food and how do you extract nutrients from there? Well, they have a highly specialized digestive system there. So another thing you're supposed to do is look at a cross-section through the animal under the microscope. And I found a picture and shared it with you there on the bottom. This is an actual specimen of a cross-section through an earthworm. And along the body wall, on the outside, you can see on the outside you have the epidermis, the outer layer, and then a protective cuticle, which is this protective covering just on the outside of the living cells, produced by the living cells. And so those things would be on the outside of the animal here, right at the very outside. And then the first layer of muscle going inward, which is this right here, that's your circular muscle. If you look close enough, you can see the strands of muscle cells going in a circular pattern all the way around. And that's going to be the ones when they contract, that squeezes down in the body cavity. It pushes on that fluid. And then the next layer of muscle is the longitudinal muscle. So those fibers are going to run along the length of the animal. And since this is a cross-section, they would be running towards us and behind the screen that you're looking at there. And then since these are have true body cavity, the space, the fluid-filled space in there, that's your coelom. Okay, and that's one of the things to know. There's your coelom there. And you move further in, you're going to find a nephridia somewhere in this location. So that might well be a nephridium there. And there's idealized nephridia or nephridium right about here. And now stoma means mouth. So this is the opening, the stoma. That would be the nephridiostome. And so stuff can enter through there and then get picked up by special cells there, and then eventually the waste products get excreted by the nephridiopore. A pore is an opening, right? So we see these. The nephridium is the organ that picks up the metabolic waste. The nephrostome is the opening, and the nephridiopore is a little opening to the outside, so the leading to little tube. Now, that you're going to see those in an actual dissection, it's going to be tough to find a specimen that was cut just perfect right through an area where you pick up all of those structures. So in our ideal model, you can identify those. In a real one, you're limited on what you could be able to identify. But there are some things you can identify. For example, the biggest structure down here, that's the dorsal nerve cord. I'm sorry, not dorsal, ventral. I ran out of space down there. So that would be your ventral nerve cord. Right there. And then there's a blood vessel right above there. That would be your ventral blood vessel. Toward the top, you're going to have your dorsal blood vessel right about there. So that would be on the other side. And this is your intestine right here. Okay. And this is the outer wall of the intestine. And that white space in the middle is the lumen of the intestine where the food passes. And you're going to notice something here is that the intestine is folded inwards. There's a little lobe-like structure that folds inwards. And any fold is going to increase surface area. So that inner fold there, hanging from the dorsal side of the intestine, is called a typhlosaur. And you can see that labeled right there in the ideal image above there. So the function of the typhlosaur, it's an inner folding of the intestine, and that increases surface area for better absorption. Right. So what have we found in these images? Make sure you can maybe try to sketch it for yourself from memory and try to identify most of these structures here. You have your ventral nerve cord, then you have your ventral blood vessel, dorsal blood vessel. We identified the intestine, the typhlosaur in there. The body, fluid-filled body cavity is the coelom. In a real image, you may not see these, right? So my best guess is that's probably part of a nephridium. You can definitely identify longitudinal and circular muscle. I did in this image here. The circular is on the outside. The epidermis is going to be the outer layer. There, I doubt you'll be able to find the cuticle or be able to say, I know that's a cuticle, but it's basically on the outside of the animal. So that's cross section, and an image like that could appear on your last lab exam. So make sure you can sketch it and try to label it overall. Maybe make a flashcard with it. And so here are questions to answer about the observations related to the dissection, observation, the cross section through the animal. So make sure you go through those and answer as many as you can, just to improve your understanding of what you've seen. And then make sure you can identify those structures, anatomical structures that we saw of the animal, external, internal, and cross-section through the animal. I'm not going to read through these because it takes too much time on the video. The last group is the Hirudinea. And the Hirudinea, again, I mentioned that these are going to lack the setae. So what are the distinguishing features of these? Well, one is the setae, the bristle structures. So the Oligochaeta earthworms have few. The Polychaetas have many. And the very exaggerated looking ones, and then the Hirudinea don't have them. There are other features that the Hirudinians have. They don't have internal walls that separate each segment, and that's something that I suppose I didn't mention. When you're dissecting through the animal, you can see the remains of what were divisions within there. There's these membranes that actually wall off every segment, and those divisions are called septa. A septum singular, so one septum, two septa. And when you're trying to open the skin, the epidermis open to get into the body cavity and go through the muscle and all that, when you get in there, you actually have to cut through the setae so that the body wall falls open. Otherwise, the body wall stays kind of hanging on by those septa. Now, let's see if we go back to this other image and we can see that separation there. Yeah, here you can see they've cut into the segments, and you can see the separations there, or is it there? Might be pointing to these separations. Those are the septums that are there going across once they completely close off little chambers within the body cavity. By the way, the Hirudinea are commonly called leeches. Their a characteristic about them is that they're ectoparasites. So they're going to steal nutrients by sucking body fluids from other animals. And their body's not rounded like an earthworm. There's more dorsally and ventrally flattened. So the backside and the belly side are kind of flattened downward. So you see, the side looks flattened in appearance. This is going to probably be the dorsal side here. Then they have two suckers. They're going to have a sucker on the posterior end. So here's probably your posterior sucker right there. And that's going to help them grab onto surfaces and move along. You can see in this image here, you can see some of the internal organs stained there in this thin section. Here's the anterior sucker there. We're going to see a video of how they move. They don't move like earthworms. Instead, they're going to inch along, kind of like a caterpillar or inchworm type movement. So Hirudinians are commonly called leeches. And again, they're ectoparasites. They feed off the body fluids, including blood of other animals. Their scientific name is a reference to the anti-blood clotting nature of proteins in their saliva. In fact, they have traditionally been used in medicine to help reduce swelling if fluid builds up in your tissues, like during an injury or after a surgery, you have injury into the blood vessels. There's an accumulation of fluid in there, and they use the leeches to actually help remove that fluid, and that actually speeds up healing, and it's actually less damaging than using a needle to try to extract the fluids from there. So there is a little section on how leeches reproduce, and basically they are hermaphrodites, and it appears that these animals produce a packet of sperm called a spermatophore. And we've seen that word before. For comes from the word phoreis, which means carry. So this packet is carrying the sperm, and each individual is going to inject the other one with a spermatophore. So the spermatophore either gets physically injected into the body cavity of the other one, and then the sperm free up, and then they go and they swim to go fertilize the eggs within the reproductive structures, the female reproductive structures. Or the packet itself is laid on the surface, depends on the species, and the packet has these digestive enzymes that help digest the body wall of the leech, and then the sperm get in that way. So it depends on the species, but that's interesting. They literally inject a packet of sperm into the other one, and then the sperm go fertilize the egg. So here is one of the activities to observe how they move, and I found a video where they're actually scooting along, and it's pretty, it would give me nightmares, especially if I saw a bunch of these chasing me. So there you can see the front end move forward and then pulls the front end backwards. And, I mean, not backwards, but forward. So first the front end moves forward by working on the hydrostatic skeleton. So you would have circular muscles similar to that, and then longitudinal muscles actually causing that bending motion there. So you still have circular motion working on the hydrostatic skeleton, and they kind of inch along. Now what's interesting is that these animals can get into the water and then they can swim, but they'll swim sideways like this, and they'll wave back and forth as they swim through there. If you look, you can find videos on YouTube. You see them actually swimming in the water. They are aquatic, or tend to be associated with moist habitats. Not always in the water. They may be in really moist habitats. So a nice moist forest that has a lot of water in there. And so yeah, there's your leech moving along there, so we can see the movement of them. I do have several diagrams to links. Check those out just to kind of look at more views of this. One of the links may be to a YouTube video to show how they move. I think these are two videos, and one of them actually shows a video of how they swim, and in an animated way, not like the slide leech, as it's locomotion along the ground there. Then you're supposed to look at preserved leech. Try to find the anterior sucker and the posterior sucker on there and observe their segment. The thing about the leeches is that although you can see the segments on the outside, inside the sections are not divided by the septa that we saw that I mentioned in the earthworms there. Another one would be activity would be to dissect leeches if we had specimens for you to dissect. We've never actually done that there, but I do provide some links where you can go check out the anatomy of leeches. But this is your final group, the Hirudinea, and there it gives you an idea of the scale. You can see the person who was filming this, or backpacking their hat there. And so now this is the end of the section, and there's questions about the leech, asking about how do leeches, the Hirudinians, compare to Oligochaetes and Polychaetes. So you'd want to write a little comparison there, make a little table of comparisons there, their different features. What are the feeding sucker is going to be on the anterior, and it had little teeth-like structures allow them to get in, cut into the body and suck out fluids there. There is no visible setae on the leech, so that's a thing that distinguishes, one of the things that distinguishes them from the other annelids that we saw. And the internal segmentation is distinct because from Oligochaetes, because there's no septa dividing one segment from the next. And then there's the question, always a question about objective one, those primary functions, right? So talked about how they reproduce. There was this little background on how they reproduce, those packet spermatophores. How does that relate to primary function? Well, that's related to primary function of reproducing, producing that next generation of leeches. And then there's a final question, 2 parts, where you're going to want to look at overall big picture associated with labs you've done so far. You did lab 37, the prior lab, which was on flatworms and roundworms, polyhelminths, and then this lab 38, there was a prior video that covered the mollusks, the phylum Mollusca, which is the clams and the octopus, and then this lab, which covered the annelids as well, so that this particular section here covers the annelids. The prior one was the mollusks, I believe, and they have true body cavities and so on. And so I think that's it for this laboratory, and this would complete a full description, relatively detailed description of what you were to do if you had those